प्यारा है। रिकॉर्ड चेंज करते रहते हो हर दिन। अच्छा कल उसका था हार्दिक माटिक का।
हेलो।
कल माटिक का फोटो ले बात
फोटो ले लो फिर वही आ गया तो लिंक ग्रुप पे
हेलो।
Good afternoon, everyone. Welcome to the 29th Annual Popli Memorial Lecture Series. We are thrilled to continue our journey today with the esteemed Dr. Gautam, Professor of Physics and Biology at Ashoka University. Continuing with the thought-provoking lecture about superconductivity that we had the opportunity to hear yesterday, today Sir will enlighten us with his journey into biophysics and share further insights into his remarkable career. Thank you all for joining us again, and let's look forward to another enriching session.
Thank you. So thank you all for returning to listen to more of my lecture. So I'm going to change from what I spoke yesterday to a completely different field, which is what physics has to do with biology. What I want to do is to tell you a little bit about the basics of biology as you might need it if you're contemplating doing or thinking about biophysics. And what are the main ideas behind, in a sense, general ideas and ways to think about biology that are relevant to physics. So there's a particularly nice talk, and I think probably an article by a person called Paul Nurse, and in which he tries to outline what he thinks are the basic ideas of biology that everyone, whether a biologist or not a biologist, really ought to know. The first idea of these is that the basic unit of life is the cell. And that's something that is so basic to the way we think about life that I'll put it out there. We will look at the consequences of this as we go along. The second big idea was that of the gene, or DNA, which contains the gene. The gene is a unit in a sense of information. And that's the first point that information turns up in the set of points that he outlined. So that's the second thing to remember. The third point is evolution by natural selection. And there is something that makes the leap from sort of molecular, cell level, gene level stuff to things that work at the level of whole organism. And the question here is, how is it if you look out of the window and you see the diversity of living organisms? Look at the garden in front of you, look at the varieties of trees and bushes and plants and flowers. Where did this come from? Why is there just one tree, one plant, one type of flower? Of course, our life would be much more boring if that was the case. But how does nature generate diversity? That's the idea behind the third point. The fourth point is biochemistry. And the fact that the rules of how life does things is really a problem in the chemistry of a certain very diverse set of compounds that are biochemical in nature. Nature isn't worried with uraniums and thoriums and all these things. There are about 20 elements of the full periodic table, about 92 of stable elements, and there are larger numbers that you can create for a very short time only. But nature doesn't concern itself with that. It's worried about carbon, nitrogen, hydrogen, oxygen, and then a bunch of other things that's much smaller. So if you take out all of the elements that are used by nature, there would be about 20 of them. It's these that combine in interesting and unusual ways to give you the biochemistry that is the basis of life. The last point is an interesting point. You might not have thought of this, and that is information. That a lot of life's purpose and what it does is really devoted to conveying information from one generation to another generation, from one organism to another organism. And life has to devise very clever ways of doing it. There are many examples. One is heredity. Of course, you are descended from your parents. You resemble them in unusual ways. Some of them may point out to you, you look just like your father or your mother or something else. There's that connection that is really information that is transferred from one generation to the other. And of course, at a more larger, more microscopic level, There's a question of how do I convey information to you? We are both organisms. How do I tell you? If I was a small one-celled organism, how do I convey to you that there is food in this direction and not in that direction? How do I myself move to it? And how do the signals that I emit convince you or tell you that this is the direction that you should also move? How do large birds coordinate? If you look out in the evening and you look at flocking birds, you'll see examples where you have large numbers of them floating in and out. How does that happen? Are birds talking to each other? Are they communicating in some way? How is it they all decide to move together in collectively? With fish, it's very, very obvious. Large schools of fish that all move together, especially when they reconfigure when they encounter a prey. How does that information actually convey? So it's interesting that information was not really a part of physics for a long time. Now you will see a lot more of it. The connection between theory of information, physics, but biologists have known this connection for a long time. But there must be ways in which biology chooses to transfer information. So remember these main points. Many of you will not see them again, many of you will have no real connection to biology. But it's good to think of these as, in a sense, the basics. And if you dive further and more deeply into these, you will understand why we chose these 5 points and no more. I'd actually like to add a 6th point, and I'll tell you why there's no more. The 6th point I will call activity. And the last sort of 5-10 minutes of this lecture where I will tell you about my own work is where I will tell you what this word means and how to understand. So right now I'm just going to put a box around it. And I will try not to delete this box so that I can come back from the question. Let's see this. Okay. So let's try and understand where physics figures in each of these things. So let's look at the first thing, what is a cell? And the first thing you need to know is there are roughly 2 types of cells. And they go by somewhat complicated names. One is called a prokaryote, and one is called a eukaryote. Every cell in your body, a bacterium, something from that plant, etc., comes into one or the other of these categories. In evolution, from the time that life actually started, so the Earth is about 14.8 to 13.6 billion years old. Life on Earth is probably around somewhere between around 4.5, 4.6 billion years ago. So we still have no idea how life started. In fact, we don't have a clear definition of life itself and what we mean. And one of the things that we might think about is what does life actually mean? How does it mean to say that something is living? If you think about that as a problem in general, that's a very hard question. All of you, myself here, are basically a set of chemicals. They're a set of atoms put together, we form molecules, the molecules interact with each other. Our final irreducible composition, that was a set of chemicals. But how is this set? But if I take these chemicals, if I take the body apart, move every atom separate, they just push it back in together. It's not going to form the body again. So there is something that is inherent in the description of something that lives as a living object that is not contained in the molecules that essentially define it. Without the molecules, of course, we cannot have a living thing. But it's not possible to argue from those molecules. In a sense, life must be something that emerges from some collective property of those molecules. But beyond that, it's very hard to say. That's one challenge for what is called synthetic biology. That to reconstitute something that lives from basic units. But that's an ultimate challenge. There are many other challenges that go long before you actually reach that particular point. Okay? So, I said 2 types of cells. These are one set, unicellular. They're organisms that survive completely on their own. They're also bacteria. Bacteria fall under this class of organisms. Eukaryotes are the cells in your body, animal cells, in general the more complex cells. Okay? Simple cells, complicated cells. That's roughly this. Let's look at what these complicated eukaryotic cells contain, because these are the cells inside your body. When we talk to the cell, we're going to draw a picture that looks like this. A picture that looks like this. It's like a circle or something more. So sometimes it can be a little more, can look like that. So the shape can vary greatly. So then it's something that is contained inside some container outside. This container is called a membrane. Membrane of particular type. And the thing that is drawn inside is the cell nucleus. And we'll come back to the nucleus and why it's okay? So that's the cell in your body, what it looks like. The first, and there are many, many different types of cells in your body. There's a big argument about how many different types of cells there are and how you can tell the difference. So right now, I mean, for a couple of years ago, it was thought there were about 240, 250 different types of cells. Now that number can be larger than 300. And people are revisiting this idea, what does it mean for one type of cell to be different from another type of cell? Just to give you one example, a nerve cell it looks a little bit like this. Okay? But again, I have nucleus here. But the nerve cell is characterized by this very, very long, long protrusion, sometimes more than that, which is called an axon. So this is what a nerve cell looks like. Other types of cells look more or less very like this, but they can be much smaller than that. So the diversity of different types of cells is also mirrored in the shapes of cells and what it looks like. And I'm not going to even illustrate the different types of complicated cells that there actually are. Just remember that there's a large number, possibly more than about 240, 250 different types of cells. Okay? And we might again use this example of why the nerve cell, what are the constraints that this imposes on the nerve cell a little later. How big are cells? Well, these sort of eukaryotic cells that I told you about are about 10 to, let's say 10 to 50 micrometers, which is 10 to 50 into 10 to the power minus 6 meters. This is too small to actually see with the naked eye. There's one type of cell that you can see. The egg cells turn out to be somewhat larger and more impressively sized. They're very possible to see with the naked eye, about 0.1 millimeter. That's a limit to what you can see with your naked eye. It's roughly the diameter of a hair, a couple of few 0.1, few times 0.1 millimeters. But by the time you get to this scale of a micron, you need at least an optical microscope in order to see that. That's the regular microscope that you might get if I do that. You can take a cell out and look at it after that. So that's the first important point I want to make is this scale of 10 to the power minus 6 meters is particularly important. Because that's the scale, as I said, if you say that biology is rooted in individual cells, this is the scale that is those individual cells must function. Okay? Alright. What does this mean for the cell? The fact that it's so small, and therefore the constituents inside that cell must be even smaller. Roughly speaking, anything of order 10 to the power minus 6 meters, called a micron, is supposed to be in Brownian region. And this scale is called a colloidal scale. Just giving you these words, might encounter them later, but roughly anything this small is supposed to be the size of a colloid in quotes. And it's also in a Brownian regime, and that idea is important to know. To tell you about the Brownian regime, I must take you back about 250, 300 years to the time of a person called Robert Brown. Robert Brown was a Scottish cleric, member of the church, who was also very interested in science. This was the time at the Netherlands in particular, where people like Van Leeuwenhoek were developing very, very useful, very, very accurate, and in a sense, absolutely first of their time, microscopes. So Mr. Brown ordered some of these, got them there, and began to look at many things under a microscope. In particular, one example he took out was pollen. So he took pollen from a plant, took it out, put it in a water solution, put it between the cover slips so that he could look at it and focus his microscope on that, and look at what is happening. So let's imagine what he saw. There is the cover slip itself, you just imagine it on a square thing. He looks at one particular piece of pollen, he separates the pollen out so that he looks at each individual grain. These grains are all about 1 micron in size. And lo and behold, what he sees is not grain here, grain here, grain here, grain here, grain here. It's something that actually looks like this. Any grain that he focuses on, or any of the grains inside his field of view, are undergoing this constant random motion. If by chance the grain came back to the point that it originally looked at, then after it would produce some completely unrelated independent trajectory that looked equally random. He found no reason for this, but at least we now know this to be what is called Brownian motion, after Mr. Robert Brown. So Brown, being a somewhat devout man, was looking for possibly religious explanation for what he did. He said, maybe there is some impression about things that I'm telling. I've extracted my pollen from a living object, a living plant that can grow in water. Maybe the pollen is moving around because it's a living object. Okay? So he said, let me test this. So let me put other things, let me take other things, let me grind them so that they're approximately the same size as my pollen grain. Let me do exactly the same experiment with a microscope. Let me ask what I see. Lo and behold, we saw exactly the same type of motion. Then he said, look, I don't know about all this stuff, I don't know whether the stuff that I ground up was, maybe there was something living inside it, I don't know. So also the time of the big expeditions to Egypt. And when the British explorers went to Egyptian sites, went to the desert, they found this huge object called a sphinx. You see pictures of the sphinx there? It's a large cat-like object that is sitting there. It's on a huge scale. So why it was built is not very clear, but it was there. And Brown said, look, this has been sitting around in the desert for, I don't know, maybe 2,000 years, 2,500 years. Surely it must be dead. There can be no doubt about the fact that this is dead. So someone who brought back pieces of the sphinx, these people were not very interested in conservation at that time. So someone just knocked off a piece of the sphinx, brought it back. He did the same thing, grinding it up very small, putting it in the microscope. Found it to be exactly the same. So Brown was forced to conclude that whatever it is had nothing to do with living or non-living. It only had to do with the size of the object that was actually moving. Okay. So Brown does this, and then for many years, nothing happens. Probably in 1700s, something, forget exactly. But that's roughly as how old it is. So little by little, people began to think about this particular problem, but they could not connect it to anything else. That was the real issue. Until, of course, the big name that out of all big names comes along. And that was the name of Einstein. And in 1905, which is what is called Einstein Annus Mirabilis, the miraculous year of Einstein, where he writes a paper on special relativity, he writes a paper on the photoelectric effect, which he was given the Nobel Prize, but he also writes a paper on Brownian motion. In a sense, the Brownian motion paper is probably among Einstein's most influential papers. Special relativity physicists were worried about things going at the speed of light. But it doesn't affect us much because we're not going anywhere near the speed of light. But Brownian motion is an archetype of many different types of fields. For example, economists and people who work in the financial market are interested in Brownian motion. For reasons that may not be clear right now, but the Einstein mathematical system of Brownian motion is important in creating whole new field. People interested in diffusion use Einstein equation. People interested in many different areas found up using the ideas that Einstein developed during his 1905 theory of Brownian motion. So what did Einstein say? Einstein said that Brownian motion actually proves the existence of atoms. And Einstein uses his theory and his observation of what is known about Brownian motion to actually derive for the first time, mathematically, the number for the Avogadro number, to estimate what the Avogadro number is. So the idea behind this is that the colloidal particle is sitting here, but around it are lots and lots and lots of other little molecules and atoms that we cannot see because they're too small to see. Under a microscope, you can see something that's about 1 micron to 10 micron in size. But you cannot see a molecule that is about 1 to 10 angstrom in size. Remember, the molecule size is about 10 to the power minus 10 meters. That's this little dot here. This thing here is about 10 to the power minus 6 meters. So there are 4 orders of magnitude that separate. This you can see under a microscope. This you cannot see under a microscope. But this does have an indirect effect. Why is that? Because these are constantly moving on their own. They're moving randomly over here, here, here. Occasionally, there will be a few more that hit this colloidal particle from this side compared to this side, in which case it will move here. In the next microsecond, millisecond, something more will hit in this direction, so it will move here. Then it will move here, then it will move here, then it will move here. And the net motion of something that you can actually see is made up of this constant random kicking by all of these other molecules that you cannot see. That's the idea behind Einstein's approach to Brownian motion. And the fact that he uses it to argue that everything is finally made up of matter is finally atomic in character. And it's interesting that you're not doing atomic physics, you're not doing nuclear physics, you're not doing things with gold foil and shooting alpha particles at them. You're not doing any of that. You're looking at something under a microscope, seeing where it's actually moving, and inferring from that all of matter's atomic character. So that takes a lot of deep thought. I mentioned this because that's exactly what happens inside the cell. Except this cell has many more complicated things inside it. Here is the cell, like this is the nucleus. There is a membrane that surrounds the cell. There's lots of other things. In fact, any picture of the cell will show you things that look like this. It will show you sort of complicated structures that have a little membrane that sits around like this. It will show you sort of dense area near the surface of the cell, which can be illustrated in this particular manner. And then there are lots of other things that are floating around. These are called organelles inside here. So, if I was teaching biology, I would spend the whole course telling you about the picture like this. I will show you a picture in the first class, and every class after that, the 20 or 30 classes after that, would be devoted to explaining what this does, 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 what is the structure of these different things. But we're not going to do that, because we're not teaching you the biology part of it. I want to tell you where physics enters and where it's important. And the first point at which it enters is precisely this point, that anything this small must be governed by ground motion. If I look at anything within the cell, I should see the same sort of random motion that I would see in the case of ground, and that happens to be true. If you look at a living cell under a microscope, you will see exactly what ground is moving around, except that because there's so much junk inside, all this other stuff, its motion is a little more constrained. It doesn't go very far. It's not as if it can wander around very far. It tends to get a little around in a more localized region rather than these. The one law that you should remember here is this is a statistical phenomenon. I can't tell what is hitting. All I have is access to the fact that it's moving around in some random way. What I can do, however, is to do a timer and say, okay, let me just do this experiment many times. In 5 seconds, if everything, all points which should have reached would be inside this circle. In 10 seconds, all points which should have reached would be inside this circle. In 20 seconds, all points which should have reached would be inside this circle. So that I can do. Okay? And the main idea here is if I take the size of that circle and square it, and I look at it as a function of time, this is the straight line. R squared is proportional to T. That's the fundamental law of diffusion. This is not the law of something that is moving with a constant velocity, because then the distance between the beginning and the end point is linear in T. R is D times T. Here, R squared is proportional to T, or R is proportional to the square root of T. That's the difference between it. It moves outward, but it certainly doesn't do it as fast as something that is moving with a constant velocity. And again, you can ask the same questions of what is happening inside the cell. Okay? The gene here is again a technical term. To do that, you have to explain that you have to go back a little bit inside what's happening inside. So inside the nucleus is, you may have heard the word DNA. You probably remember this what a DNA molecule is. The DNA molecule is basically a double-stranded object which has a helical structure. And this is the helix, the right-hand helix. And this is for DNA. And those of you who remember what the description of the DNA is, will remember that it is formed by pairing bases on these two strands. So it's like you take a ladder, the rung is the side of the ladder is this thing here. You connect the rung from one to here, and then you twist it around in order to get it. So this is the double helical structure of DNA. DNA can be a large structure. Okay, how large? Guess for me how many cells a human body has. My body, your body. Give me a rough estimate in terms of 10 to the something. Just guess.
10 to the 13.
Huh?
10 to the 13.
10 to the 13 is right. I hope you didn't look it up. It's a very good thing to remember. That's a nice number. That's 10 trillion. So there are 10 to the 13 cells inside your body. Okay. I said the cell nucleus inside there contains DNA. In fact, most of your cells contain 23 pairs of DNA molecules. And these are in the form of chromosomes. So the chromosomes are labeled 1 all the way up to 22. And then the X chromosome or Y chromosome. That's just a detail that we need to worry about. But each nucleus of a cell contains 23 pairs. Let me take this pair out, and let me imagine taking this here, attaching it at the ends to form one large molecule. This itself is a large molecule, one of these you see. I imagine taking out each unit from each nucleus and attaching it. How big do you think that is? If I just, how long would that be? Again, you can guess.
More than half circumference.
Huh?
More than half circumference.
No, that's too large.
Radius?
Huh?
More than half radius?
That's too large. It's something more reasonable.
2 meters.
2 meters. That's again correct. Very good. Okay. Alright, so multiply 2 meters by 10 to the 13 cells. That's 2 into 10 to the 13 in length. So if I took from each nucleus, I took all the DNA attached to it, second nucleus, all the DNA attached to it, 30 to the how much is that? What sort of length is that? Compared to again, last guess that I'm asking you to make. And you should not reply.
2 trillion meters.
This guess doesn't matter. So it's roughly 10 to the 13 meters.
It's 10 to the 8 meters.
Yes.
Or 3 into 10 to the 8.
Yes. So what does it compare to? 3 to 10 to 8 meters is something that you should remember. It sort of corresponds to something.
Right.
Huh? That's right. How far light travels in 1 second is 228. It's 2.99 if you want to release that from 9.8 something. Okay, I'll sort of cut it short and tell you. This is enough to go from Earth to the Sun almost 100 times. Now whether the factor of 2 etc. is correct, how exactly you do. So it's huge. So inside every one of your body, you're carrying a length of molecule. Of course, we said we have to tie it together as a that's a minor point. That's equal to something that can go from the Earth to the Sun and back 6,100 times. That's how tightly. But even this 2 meters is pushed up into a size that is less than 1 micron. Maybe it's about in this example, typically it's about 1 micron. You must put 2 meters inside 1. 2 meters is about this much. 1 micron is smaller than I can actually see with my naked eye. I can barely see it with an optical microscope. So that's an incredible feat of packaging to take something that is 2 meters in size and squish it into a region that is about a micron in size. That's one thing to do. Also ask how does the cell do that? How does the cell actually manage to package things inside? Okay. So stuff inside the cell is constantly moving. And in fact, this is a barrier for the cell to actually do anything. It can't do anything purposeful. It's constantly being asked to move here, there, etc. random. Because some random set of molecules is coming colliding with it. So how does life surmount the fact that it's constantly assailed, constantly being attacked by Brownian motion, which is in essence random, for pushing you from side to side in a random manner. So that's where Brownian motion enters. We've already got to some sort of description here. We've talked about DNA. I can tell you about one physics example that people are interested in for DNA. You can do clever experiments where you take a substrate or wall here, take a DNA molecule like this, and at the end of the DNA molecule, you attach a colloidal particle, about 1 micron in size. You can take the colloidal particle and create an arrangement of lasers. It's called an optical track that tracks it here. Okay. Now I can measure this distance right up to the center of the laser of the colloidal particle very accurately. I can also measure the force that I'm exerting by moving this trap to the right hand side. What is the relationship between the force and the extension of this molecule? This is an experiment that miraculously can be done. These are called single molecule experiments. And now this has been developed to a fine art But Nobel Prizes were given out for these sorts of experiments a couple of years ago. Steve Chu was the person, a bunch of others who actually showed Arthur Ashton, Steve Chu, blah, 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 these are the people who showed that these experiments could be done. And now they give you very precise ways of actually manipulating molecules, pulling them, stretching them, twisting them, etc., to understand how molecules behave. And it's particularly important in biological systems because what you do, their function depends in a sense on the conformation and shape that they can actually take. That's again important. So that's one point where physics centers. Here's an experiment, and here is a theory that you're required to develop for this experiment. Right now, the theories for these are so accurate and believed to be right. They used to benchmark the experiments. That if you want to try, look, is the thing that I bought for $1 million from the company that allows me to do single molecule experiment, is it working or not? I would actually calculate. I would actually do a force extension experiment and put it into the formula that I use for calculating force extension and see that it fits. And if it fits, then the equipment works. This is an interesting way of doing things together. So we've talked about cells, we've talked about genes. Genes are parts inside the DNA. So if I look at the DNA, if I pull the DNA apart, I can say that look, it's supposed to be base pair. And I know that it has A here, it back pair with the T, if I have G here, it pairs with the C. So finally I have some sequence that looks like this. These are bases: adenine, thymine, thymine, guanine, and cytosine. These are 4 basic building blocks of DNA. These are the little rungs inside the lab here. And the fact that these rungs consist of paired bases is something that was established through biochemistry in the early 1950s, early 1950s. But the DNA, the structure of the DNA, double bases and how it enters this double helical structure, that of course was Watson and Crick, who worked this out in and got Nobel Prize in the 1950s, early 1960s. That again is a huge development where people understood how heredity actually happens. So the mystery of how you resemble your parents is a mystery that was solved by Watson and Crick, who understood how the DNA structure contains the information that you require to pass from generation to generation. And all of you have a genome. If I could took every bit of DNA from your cell, so you take a typical cell, take the DNA out of it, identify each chromosome, the 23 pairs of chromosomes, take one of the 23, each of them corresponds to a sequence. I only need to tell you one part of this because automatically the other part is fixed. A must pair with B, G must pair with C. So this whole line of characters and how it's represented in your body is your genetic code. That's your genome, technically speaking. It's your genome. And the culmination of few decades of work was finding out what the human genome is. It was a human genome project which came to fruition in the early 2000s. Then we actually figured out here's the representative genome for everybody in the world. That representative contains some amount of genes. It's an average over many people, they wouldn't know, so average over 100 different types of people. But of course, what makes you, and him, and her, is small changes in the gene. We were exactly the same, exactly the same genome, and the same thing that went into it. Then of course, we would be very, very largely similar to each other. A little more layer of what's called regulation is just about that, but mostly, most of the information that we need is actually contained in that sequence in the genome. So now we're interested in small differences in the genome that can distinguish one person from another, someone from Africa, someone from India. We can now use this to determine how populations move. We understand now that human beings really evolved out of Africa. It was a small region within Africa that gave rise to every human in the world. There were many streams by which they came out. They first came out to Africa, to parts of Central Asia. They led one sort of round in which they came to India, came to the east, south, and then they moved on to the Far East. Then there were subsequent ways that people came back from that side, from Australia, from Far East, came back here. Another group of people came from here. People call them invasions, but they're hardly invasions. People were just moving wherever they wanted to at that time. And we are a composite, a mixture of all of these people. So Tony Joseph has a nice word for this. So they're not pizza. They're not pizza toppings. There's some base that's common. And so somebody can have tuna, chicken, and broccoli or stuff on their pizza. These are minor differences. The main base really comes from all of what happens, the whole sequence of migration from many years before. So we're interested in molecules like this, their structure, forces we could apply to them. That's one important that we could do. I'm not going to talk about either of these two particularly, because that would take another talk on its own to talk about the natural selection, the nature of evolution, etc. And biochemistry is not really the topic of the physics discussion, and that needs a lot more background into the area. I want to talk about information and activity right now. Because the activity part is something that most represents my own work in this particular area. So first of all, information. And that's where biology now begins to talk to other fields. The understanding of how biological information is copied was a big breakthrough. And that's again something that was hinted at by Watson and Crick's work. But the machinery that led to the copying of the DNA was something that was understood some years later. The fact that you could take a DNA molecule and copy it, that to how a mother's sperm and the mother's egg and the father's sperm could come together and then divide, reproduce and form something that was complicated as well. Remember again that all of you started off with one cell. Now you are 10 trillion cells. That's 13 orders of magnitude you have changed in terms of number. And the process of that is called development. And that's again a huge area of study. Both mainly within biology, but also the physics of development. What are the forces that must act when something grows? And again, I can recommend very strongly that you look at videos online of how a single cell being doubles, triples, forms an embryo. The incredibly complicated thing which you can now look at through taking a long movie of several hours and days and compressing it into something like 2 or 3 minutes of visual, so that you can actually see each of these different stages and how they actually happen. Okay? Information we now understand is the point where biology talks to computer science and biology also talks to physics. Physics is in a sense the king of information theory. When Shannon came along and tried to understand how much information is there in a sequence, the equation that he turned to was the equation for entropy that had been derived 50 to 100 years earlier. So the form of entropy that was written down by Gautam. He wrote it in a more mathematical form, KB didn't turn up and so on. But those ideas really are much earlier like that. So that entropy is something systems try to maximize, and the entropy is something that governs the nature of the information that can be carried in a particular signal. And that's where information theory, communications, all of the things to play their role, and connect on to the way information is transferred by other processes. Okay. So let me now begin to talk about activity. And when I tell you about activity, let me tell you about one piece of biology that I find very interesting. And that is, when I do this picture, I do these sort of long stick-like objects. Okay, you may not have noticed when I do it because there were a lot of complications with that. But this stick-like object is called a microtubule. Microtubule is like a long stick that I showed you there. It has a direction, in the sense it's made up of units called A and B. It doesn't matter what these are. But the combination of A and B together, lined up like this, is what gives a microtubule. It's actually more complicated. It's a hollow structure, it's a helical structure, but roughly there is an inherent direction to this based on the sequence of A to B and where B follows A. Okay, that's a detailed view of it. But now let me tell you something. There are molecules that associate with the microtubule. And I can show you what they look like. They look a little bit like this. These are called heads. This is called cargo. And that's what that molecule is. This molecule is called a molecular motor. It's called a molecular motor because it does something funny. It carries a cargo. Think of someone who's a porter in a railway station. The porter is carrying a big bag, he's carrying a luggage full. That's like the molecular motor. And the porter is moving in one direction. You have got off the train at one corner, the last bogie in the train. The exit is over there. And you ask the porter to carry it to you, the porter is walking down. There are molecular motors that walk in this direction. And there are other different molecular motors that walk in the opposite direction. There are some motors that come towards you and some motors who take luggage away from you. And they're very specific. The same porter does not do both things. Okay? So the porter is here carrying cargo, moving along. A little later it sits here. A little later sits here. And you can look at this under a microscope. You can actually track. This is very small. So what is normally done is You add what's called a fluorescent label to that, and then you look at the motion of this label in time. And you find that it sort of moves like this and it stops, and it moves like this and it stops. But more or less moving in one direction, from this end to this end in the case of the protocol. You should immediately tell me that, look, how can this happen? Because this is a Brownian machine. The only thing that I know about is that it's pushing it there, pushing it there, and there's no sense of direction in any of these pictures that I showed you. How can something that small, how can a molecule that small move unidirectionally when the forces that it is experiencing have no direction? That's a basic question in physics. That is, you can understand everything that I told you. It's an absolutely central question. There is nothing in the physics that we know so far that will tell you how a molecule can move in one direction. If there are forces acting upon it, if I put electric field here, the molecule is charged, then of course it will move in that direction. But there are no electric fields, certainly none that we know of, that will give you such large scale motion that starts off here and winds up here. And other molecules are not doing that. Other molecules are doing happy random motion that is there between parts. So the understanding of this is called a ratchet. It comes from the ratchet and the ball experiment. A good place to read about this is Feynman, in the Feynman lecture, who explains some of the physics behind this. The translation of this idea into biology was achieved in the early 90s. In fact, although the ideas are very old, they go back to Curie. But the implementation of this and why these are relevant to these particular molecular methods came to be the early 90s. I'll just tell you I'll give you a hint of the answer of why that's true. For this molecule to move, for the molecular motor to move, here is the attachment to the cargo, here is what is happening. It needs help. It needs help in the form of another molecule. It needs help in the form of another molecule called an ATP molecule. So this molecular motor has one particular location, it is called a binding site, where the ATP molecule comes and sits. Once it comes and sits, it decays, it breaks the ATP bond and becomes an ADP, the triphosphate bond becomes a diphosphate. When the bond breaks, it releases energy. The energy that is released goes into the molecule, raising its leg, or head in this case, and moving it forward. The construction of this molecule is such that it can only move forward and not backward. At the end of this cycle, the molecule has ADP, no longer has ATP, and then the next last step in terms of throwing out the ADP, so that it reveals that binding site once more. What secret do we learn from here? That it's not just thermal fluctuations and Brownian motion. There is some external help. It needs another molecule. What is more, it needs the concentration of that molecule to be out of equilibrium, because it needs to constantly take in ATP and give out ADP. And somewhere else there must be something that takes the ADP and makes it back to ATP. We won't talk about that. But that is a central point, that biological systems, unlike anything that you may have heard of or studied so far in undergraduate physics class, are systems that are out of equilibrium. And they're out of equilibrium in a very specific way, because the amount of ATP molecules that are used to pass energy through the system are held out of equilibrium. Okay. So now to take that and translate that into physics, a more complicated question, which we won't do. I just wanted to tell you what that is. But I want in the last 5 minutes or so of this talk to tell you where my own work combines this particular aspect. The fact that nature, living systems are out of equilibrium, the fact that you must pass energy through them, the fact that these small molecules of ATP are needed that have to be broken down. They're like the food. The cell processes consume food in the form of ATP molecules. So it's going to constantly feeding them. And therefore, the flux of energy must be maintained through the cell, which keeps it out of equilibrium. That idea here is the idea that the last word that I wrote down, which is activity. Such systems which are fed energy locally and consume energy are called active systems. So now I want to go back to this question of what happens inside the nucleus. And for that, I suppose this little example here. Here is your cell, here is the nucleus, and here are your chromosomes. There are small chromosomes and large chromosomes. As I said, I mentioned the numbering here. I said they are numbered 1 to 22 and X and Y. But this numbering is roughly in the order of longer to shorter. So 1 is the largest chromosome and 22 is the smallest chromosome, roughly speaking. So now the question is, suppose I tell you this is chromosome 17. And this might be chromosome 18, this might be chromosome 22, or this chromosome 2, etc. I can identify that instantly. In fact, if you suspect that if an infected mother suspects that their child may have a genetic illness that's related to the number of chromosomes, they can go and get this test done. It just counts the number of chromosomes and make sure that they're all okay in terms of their sizes, structures, and so on. It's called genetic karyotyping. It's done in a fairly standard way. Now the question is there anything special about the way these chromosomes are placed? Am I equally likely to find chemicals here, as here, as here, as here, as here, as here? Is there any logic to where the chromosomes actually appear? You might first think that look, everything is brown and everything will be brown like this. There should be no particular logic. I can find 22 flows here, I can find it towards the outside, I can find it somewhere else. It doesn't really matter. If I did an experiment, I would find no difference between positioning of any of these chromosomes. That turns out not to be true in an interesting way. If I ask what is the probability of finding a chromosome at some distance r from the center of the nucleus, and this is one level of the boundary of the nucleus, I can generate a probability distribution that looks like this. Okay? This says that look, I'm not likely to find it here, I'm not likely to find it here, and somewhere in the middle I'm quite likely to find it. That's roughly what it says. It's a little more complicated than we actually multiply the factor of 4 by r squared, etc. But I'm just giving you the approximate idea. And if I did this for chromosomes, if you say chromosome 18 and 19, I will find that chromosome 19 is here and chromosome 18 is here. They have very distinct distribution. I'm much more likely to find chromosome 19 closer to the nucleus than I am to find chromosome 18. I can do this for chromosomes 1, 12, whichever one you want. I can generate a picture like this. This is obtained by taking lots of pictures of nuclei, tagging them with a fluorescent laser, finding out where the center of mass is, doing this 200, 500, 600 times, and then plotting it. Making a histogram and plotting the histogram. That's essentially what goes with it. But the real miracle, the real surprise is that these are not the same. There's nothing that can tell us why they should be the same. We just assume what we know. That they are the subject of Brownian motion. These are molecules, raw molecules that can move around. And therefore, they should all have been approximately the same. So my own work has been devoted to understanding exactly what this difference is. And what the way that proceeds is to say that activity is really important to determine. How does activity figure? Because chromosome 18 is basically a DNA molecule like this. The DNA molecule contains genes over there. The genes have a function, so the genes are being written out constantly. The more genes there are in a particular region, the more active it is. Because machinery that does things, that converts DNA into RNA, is a machine that also eats up ATP all the time. Because it's eating up ATP all the time, this is no longer an equilibrium system, it's a very non-equilibrium system. Not only that, the non-equilibrium is distributed very unevenly along the whole chain. So then the guess is that if I could simulate this, keeping in mind where the genes are, keeping in mind that in a particular type of cell the genes are being written out more here versus here versus here, I should be able to find out the secret of why these different chromosomes have different distributions. That's essentially what I've been doing for the last 12 years. The problem of biophysics, to understand what the basic idea is and how is it that we can develop actual calculational tools using simulation that can describe the distribution of different chromosomes. So this takes a very complicated problem. The whole messy interior of the nucleus was solved. It takes in two ideas from physics. One is that these are molecules, long molecules, that does polymers, which you can then simulate. But these are not simple polymers that you might look at in other contexts. They're non-equilibrium polymers. And they're non-equilibrium in a very particular way because they have little genes inside them, which we know, because we know the human genome. We know what genes are being written out to make this type of cell. When that information goes in, together with the information about non-equilibrium, to try and understand how to predict these structures. Then we can ask a whole lot of other questions. What are these are called chromosome territory. The shape that chromosome takes are called territory. What determines how rough these territories are? Are they more spherical? Are they more elliptical in nature? All of this we can now address using these methods that I outlined here. And again, this emphasizes the fact that life is finally a non-equilibrium system. It relies on activity. Activity is the consumption of ATP molecules at a microscopic local level, You must constantly keep making the ATP molecules and break. In fact, the human body breaks down roughly half its weight in ATP every day. That's how important that molecule is for you. A bunch of that consumption happens there. 20 to 30% happens in your brain. So your brain is constant. The ferment type of activity. You cannot understand life without understanding activity. This is one example of how physical ideas of body activity actually manifests itself in things that can be measured and calculated. But there are many other very nice examples. It's a very active, involved area of physics, which is just beginning to address that question of what is life. Thank you.
Thank you all for joining us today as we conclude day 2 of the Popli Memorial Lectures. We are incredibly grateful to Dr. Gautam for continuing to share his profound experiences with us and inspiring us with his dedication to his field. As we wrap up, I'd like to firstly thank everyone who joined us today, and we also thank the Photography Society for covering the event. Now, we look forward to welcoming you all back tomorrow for the final lecture of the series. Now, I'd like to request all of you to kindly step outside for the group.
Yes, you can ask questions.
So, you talked about the probability of finding genomes. During the birth, when there's a single cell, does that affect the mental state or the neurotic state of the baby that's being born? For example, autism, Down syndrome. Such things can be affected by this genome, by this chromosome that's being found at different position or a missing chromosome.
That's a very hard question. And in principle, you're not even getting to the sort of nerve cells. Finally, autism is supposed to be phenomenal because that's very neural in character. Some nerves behave differently from what they would do in normal situations. But right now, when you're born, it's really basically one cell. That is a potential for becoming, after division, many other cells. Is there a special genetics of autism? Are there special genes that are mutated in specific ways? We're not completely sure about that. That's my understanding. It's not my field. There's probably many genes, subtle changes in many genes are involved. Maybe the environment is also involved. Maybe if you put the same person with the same set of genes and put them in a different environment, they would not become autistic. We don't know. We really don't know.
A question about the same thing. Is the probability distribution just purely statistical? It's like the heavier chromosomes tend to want to have more degrees of freedom, and it just stays in the center, while the lighter are pushed out.
So that's one way of looking at it. They're distributed by size.
Yeah.
That doesn't turn out to be really the case because chromosomes 18 and 19 are roughly the same size. But they're very different in the number of genes that they contain. 19 contains many more genes than 18. So the idea is the number of genes per unit length, so the gene density, is what is really crucial in making this. Even if you take chromosomes with very different lengths of the same gene density, then for example, there's a chromosome 12 and 20, I think, that combination. Then this turns out to be quite similar. That suggests that gene density is what is differentiating the different types. And that size doesn't have too much to do with it. Because it's complicated because experiments are done on many different types of cells. Some of them are more squished types, some of them are more spherical. And how does the squishing, the shapes of the surrounding nucleus have an impact? It's a more complicated question. We have studied some of that. It's also a question of how we interpret experiments. But the broad idea is that it's gene density, the number of genes divided by the length of the chromosome, that contains the difference between things like this. The more genes you have per unit length, the more chances there are of them being transcribed. So when we that require the consumption of energy. It's the consumption of energy locally that makes a difference.
Yes, and one analog, I don't know if it is the same effect. So if you have a box of, I don't know what's the word, mix bhujia with nuts in it, just shake it a little when the nuts will come up. Is it like the same effect?
So that's called the Brazil nut effect. And sort of variants of that. It's not real, that's very size dependent and that's very granular. I mean, it's basically temperature is relevant. It's the fact that you're shaking it over here. Here, temperature is relevant. It's just that activity adds to the temperature in order to give you the stability. If only temperature was there, then you would have some sort of distribution by size, roughly speaking. But activity overrides that and gives you this very specific difference between activity that you cannot explain otherwise. That's a good question. That really means It's not completely correct to think of it like that, but that's also shaping it is creating a non-equilibrium system, so providing energy to it from the outside. So in that sense, there is a relationship between the way this formulation is that.
My question related to health. You talk about ATP. Is that the only molecule that's involved in this? That's question number one. And question number two is how well can you reproduce the shapes of this curve?
Okay, first question is ATP is the one that is by far the most important.
But
In general, GTP is another one that in other types of processes that become important. But for the ones that we are considering, it's really ATP that's completely dominant. And in fact, in most of the processes, it really is ATP and nothing else. And the second one was how accurately can you do the answer is very accurate. But accounting for the fact that the experiments themselves have error bars, but those error bars certainly will tell you to do the separated out. But not only can we do this for 19 and 18, we can do this for 12, 20, any chromosome that you want. You can generate similar quality of cells. What is more, you can solve this very interesting and old problem. Why is it that the two X chromosomes in women are differently rotated? So quick background to that. Women have two X chromosomes, the man has an X and a Y. If women have two X chromosomes, but if each of those chromosomes is making as much protein as the other one, then they would have too many proteins compared to human beings compared to men. And therefore that would not be good, there's a deleterious effect. So miraculously, one is switched off. In every cell of a woman, one X chromosome is switched off. It's called inactivated. But others have identical sequence. It's just that there are things that attach to them that make them less active and not able to do anything. Whereas the other active one functions as usual. Active and inactive X and female cells are located differently. There's no reason why that might really happen because as I said, they're identical in sequence. There's some biochemical change that happens to them. But the difference of where they position that is very substantial. So what we showed is that the X chromosome, active X chromosome must be inside, the inactive X chromosome must be outside. And this is a process, an observation that dates back 100 years.
Experimentally, how do you figure out that within a living cell that that one of them is active, the other is inactive?
You can label the genes and products of the genes that the others come out. If one keeps generating a product, keeps generating RNA, the other doesn't.
And you can monitor that.
You can monitor that in a living cell. You can take this out and look at it in you can do various other types of biochemical stuff. That's completely established. And in fact, it's a beautiful problem in biology. How is it that one of these, and only one is switched off? And not only the accident, the other one. How do you switch off two identical looking molecules to start out with? How do you make sure that one is completely suppressed and one survives in a cell? How is that choice made? When is it made? And what are the consequences?
So when is it made? How far from formation of the cell? How far down is that choice made?
That's an interesting question. I don't know the answer to that. When it's made? I mean, the cell divides, and if you have mother cell dividing progeny, the cell must duplicate its DNA.
Right.
And does that contain little markers that tell you this is the one that in the daughter cell is going to be inactivated or activated? I don't know. It probably does. So probably even the mother cell, the mother has already marked the copy that is going to be inactivated in the daughter. That's the only explanation that I have. I should look it up and see what the right answer is.
It's a related question. How does the cell decide when to divide?
Oh, great question. And the cells have a little timer inside them, that's a biochemical timer, that basically is determined by the balance of biochemical elements inside them. If they reach a certain level, genes are being switched on, and then begin the process of cell division. So how that happens is a whole separate question. In fact, Paul Nurse, who I mentioned there, was one of the pioneers of discovering how this clock is beating. When cells divide.
I think that typically you think of life as a very, right from the cell, the ecosystem, very organized. Organization is not there in this.
In this picture.
The 6 points which you wrote.
That's true. I suppose it's their information in a sense. That information is contained in spatial organization and in spatial relationship in what there is. But it's not, it's in a sense that's emergent and at a higher level. Already when you said the cell, you said that There's organization inside that I'm not talking about. I'm just saying this is the fundamental unit of life. But you're right, it doesn't figure as a specific organizing principle. Partly because I think you can limit that organization in other ways, in non-living systems. You can create instabilities that are very precise, that are, or create reactions like Wilson's you pattern that move on. There are some things that look very ordered, they generate order spontaneously at a large scale. From this simple thing that you can put together in ordinary laboratory and have nothing to do with anything living. So this is a subtle question. Right? No more questions? I'm around tomorrow too, so you can ask me questions about lecture 1, 2, and 3 at the end of the day.
Now I'd like to invite outside for a group photo and refreshments.